So, yes, my name is Zehra, and I will today talk about a part of my PhD project. Okay, so we know that all of our ancestors originated in sub-Saharan Africa, and from there they migrated over all the other continents. So this is something that we know, and there's different ways of studying these migration patterns, like using archaeological findings, for example. But what we use is our genetic differences. So we know that when people live and migrate, they obtain genetic differences, and only the genetic differences of those individuals that successfully reproduce can be inherited over generations. So they become more frequent. So this means that we can correlate migration pathways to these genetic differences, to the respective populations, and to a time range. And the genetic differences that we find to be very interesting are genetic differences in the Y chromosome, because the Y chromosome is nearly entirely inherited from or transmitted from the fathers to the sons. But this, of course, also means by looking at only the Y chromosome, we are actually looking at male migration patterns. And these genetic differences that share a more recent common ancestor, they can be grouped together. And for this group of genetic differences in America, we speak of haplogroup Q, this is the name. And specifically in South America, the group is called haplogroup QM3, which is just a subgroup of haplogroup Q. And these genetic differences can be put into this tree based on their differences and similarities. So we speak of a phylogenetic tree here. And now the problem in South America is that we actually don't know how the first settlers of South America actually migrated. And this is because for the Y chromosome, there is a lack of continuous data. And this is because of the European occupation in the late 15th century. And so a lot of Europeans coming into South America and a lot of African slaves being brought into the continent. And this led to a lot of native men to live in small, isolated populations. And what happens when people live in small, isolated populations is that they lose a lot of their genetic variation. And then there was also men that were of Native American ancestry that actually reproduced with European and African women. And the children of these men are called admixed South Americans, but they have a native male lineage. And because these individuals, they did not live in small isolated groups, but they admixed with this, and they lived in big populations and it mixed with the foreigners there, these individuals could retain a lot of their genetic variation. So the aims of this project were to find genetic differences in these admixed South American men that have the native male lineage, so this is important. And we wanted to determine relationships among these genetic differences. Then we want to find geographic patterns of the genetic differences, and based on these correlations, predict migration patterns of the first settlers of South America. So what we did in the study was that we sequenced the majority of the Y chromosome of 64 individuals from these regions. And we used a method called hybridization capture, which is a next generation sequencing approach where we have our DNA sequences and then we use RNA baits. So this is the pink short sequences. And they are complementary to the regions that we're interested in the Y chromosome. So they can be retained, and all of the rest that is not interesting for us can be washed away. So we only sequence the interesting DNA sequences using massively parallel sequencing. And this 64 genetic data was supplemented with data from like publicly available data from 211 people. So these are whole genome sequencing data that are ancient or modern, that are of indigenous groups and also admixed natives. And now we have 275 genetic data. So for the bioinformatic pipeline, what we are doing is, so we start with these DNA sequences that have these artificial sequences attached. So we have the sample index that is, we know that is unique to the sample. We have the adapter and then the molecular barcode. So the molecular barcode is like a unique molecular identifier, which we heard yesterday about. So these are short reads that are, or short sequences that are unique to this specific DNA molecule. So what we did first is that we separate these DNA sequences based on the sample index, and we trim the sample index. Then we trim the adapter and molecular barcodes. We align this DNA sequence to a reference, and then we know that this is a forward read, for example. And then we make use of the molecular barcode. So we compare all the DNA sequences that have the same molecular barcode. So they originated from the same original DNA molecule, and they have the same orientation. So they have all the forward, they're all forward strands, and they came from one original DNA molecule. And because these are all DNA copies, they should be identical. So whenever we see, like in purple, that there is a difference, we know that this is an artifact that was introduced by PCR, so by the amplification. So what we do then is we do the single strand consensus sequence deduplication, and this just means to compile all of these copies to one strand for the forward read that only contains variants that are in common in all of these copies. So simultaneously, we did the same for the reverse strand, and then we do the duplex consensus sequence deduplication. So we make use of the consensus reads for the forward direction and reverse direction, and we align those. And now in green, we see that there is something that is not in common, but the forward and reverse strand, they also originate from one DNA molecule. So they are the two original strands, or the two strands from one original DNA molecule. So we remove this possible artifact, and then we only keep sequences of the same region within the Y chromosome that is covered by this duplex consensus of at least two different original DNA molecules. So the one in the light gray is from a different original DNA molecule, and this is to prevent that we misinterpret a mutation that was found in only one DNA molecule as being reflective of the whole genome of this person. So the whole purpose of this and the molecular barcode is to reduce the error rate. So then eventually we can do a base quality recalibration and call a variant that we can be sure of that it's true. So in these 64 samples where I did the laboratory work, we found nearly 13,000 total variants. The majority of those were SNPs, and around 10,000 were found in only one individual each. So of all these 12,000 SNPs, 7,500 were not previously annotated before, and the rest was in databases and other publications. And then the remaining 820 variants were insertions and deletions, with the majority being novel again, and then some that were annotated before. When we add on these 211 and 64 samples that were from the literature data, we find like nearly a tenfold increase, or 110%. So it's 125,000 total variants with 118,000 SNPs, and then overall 80,000 were not annotated before. So when we try to put these SNPs, now starting with the SNPs that were only found in the 64 individuals, when we try to put them into context relative to the lineage that we're interested in, the QM3 lineage, we can look at this pedigree or this phylogenetic tree on the right side. And let's say this red mutation is the mutation that introduces the QM3 lineage. Then we speak of all of the variations that follow, including this red one, that belong to the QM3 lineage. So out of these 12,000 SNPs, we found 10,500 SNPs that were only found in individuals that belong to this QM3 lineage. Then 690 SNPs that were only found in individuals that specifically do not belong to individuals of the QM3 lineage. And then there were also 1,000 that were shared. So they were found in individuals belonging to this lineage as well as not belonging to the lineage. And then for all of the individuals, we looked at 96,000 of the 118,000 SNPs were were found only in individuals of the QM3 lineage. 11,500 only in individuals that do not belong to the QM3 lineage, and then 10,000 that were shared by both groups. So overall, we were able to find 120,000 variants when including the literature data, and those meant that we found 80,000 novel SNPs. We were able to put them into relation compared to the QM3 lineage, but now we want to search for geographic patterns of these genetic differences in as many samples as possible in order to predict migration patterns. So this work was only made possible due to the help of my supervisors, Professor Vanya Pereira and Klaus Börsting from the University of Copenhagen, Denmark, and Professor Leonard Guzmão from the State University of Rio de Janeiro, Brazil, and with the help of 11 other groups all over South America. So I'm asking now if anyone is interested, please contact me. You can use this link, this QR link, if you want to be part of this project. Thank you. If you have any questions.
Well, Sarah, I have a question. Your procedure reminds me the 16S purifying in metavar coding, when you are really trying to find single variants. I don't know if you know it, but I'm just curious if it's very similar or if you find some difference. And my other question is, why do you think is so relevant to study these variations? These haplotypes?
Yes. So this is the main haplogroup in South America. So if we want to find out how the native settlers actually migrated, this is the haplogroup that we have to look into. And we need to find these variations because right now we had published a work that was finding like the frequencies of different haplogroups in these countries. And we found that most individuals belong to the QM3 lineage. So based on this data, we cannot say how the people migrated because it's just putting everyone into one group. So we need to find a higher resolution within the haplogroup QM3 lineage, so we can find some patterns based on these correlations in order to say how the people migrate, because this is something that is not known still.
Maybe you can answer me after the meeting about the 16S question, because there is another question for the public.
Great, I like the 16S question. Your migration hypothesis map shows mostly north to south migration with some infusion from Europe. And I'm wondering, will you be looking for, or have you found south to north, perhaps indicating some Asian influx in the Southern Pacific regions?
So the figures that I was showing was just to explain the pathways. What is known is, or what is suggested is that there has to be a migration along the Pacific coast, which also makes sense because also within North America, people are suspected to have migrated along the coast and also between ice sheets. So people need to take into consideration a lot of information, not only genetic information, but just based on this kind of information, I can also say that there is suggested to have been a migration pathway from the Pacific coast in the direction of Brazil based on a decrease of genetic variation. So there's some suggestions, but there is no clear answer. But yeah, and then you were asking about Asia. So the people have in North America, the natives, they have definitely migrated over Siberia, over the land bridge that used to be a land bridge. This is also how this haplogroup Q was introduced into the continent. So haplogroup Q originated from East Asia. So this is where the origin of the natives are.
Sorry if I'm showing my ignorance here, but maybe I've seen this kind of thing done with mitochondrial DNA from the maternal lineage before. And I'm just wondering, looking at the Y chromosome, what kind of different patterns can you see?
So, so we're now comparing male and female migration. There is often a similarity at worldwide scene. They have different haplogroup names for the mitochondria, like we have heard earlier before, today before. But there's some similarities worldwide. But when we look at South America, there's a difference between women and men, like native women and men, because the women, they reproduce more successfully in the continent, and it was the men that were like excluded from reproduction. So historically, there is this difference, but based on mitochondrial data, this is what I was answering, these pathways along the Pacific coast, this is taking into consideration female migration, because the male migration, it's a complicated one, because now looking at South American populations, of course it depends which country I go to, but if I go to Brazil, in specific regions, it's mostly African individuals, so it's not a high percentage of native men. So there's not been so many studies that have been covering different countries. So there's in one country there's some studies on native men, but not so much taking the whole continent into consideration. But there's some parallels between male and female migration.